Microscale thermophoresis is a technique used for quantitative analysis of protein interactions. Basically, any type of biomolecule can be tested using this instrument. Proteins with other proteins, liposomes, DNA, and even small molecules. Due to the high sensitivity of this instrument, it does not really matter how big the weight ratio of the two interacting partners is. Measuring is even possible in complex bioliquids like cell lysate. Therefore, we do not have to use ideal solutions that do not even come close to the real conditions. Before looking at how this instrument works, I'll tell you what binding does to a molecule. Binding can induce many changes, be it in size, charge, size of hydration shell, or conformational changes. So, the ability of a molecule to move through a solution changes after binding with another molecule. This is the main factor that we take into consider. In MST, the mobility through a temperature gradient, or in other words, the ability of a molecule to move through a temperature gradient is investigated. In this instrument, there is an IR source and it heats up the sample. As a result, the molecules move away from the heated spot. Unbound molecules move fast and bound molecules move slow, just like a person carrying a very heavy bag. Molecular flow along the temperature gradient is opposed by mass diffusion. This is like Newton's first law. Objects do not like to change their state if they don't have to. After some time, both effects balance and this is called the steady state. But how can we image this motion? In MST, one binding partner is fluorescently labeled. That means we simply introduce a dye. These fluorescent materials are covalently attached to the molecule of interest. We must make sure that the dye does not interfere with the original binding site. Therefore, the proteins are labeled far from the binding site, minimizing the disturbance to the interaction between the biomolecules that we are testing. When molecules have intrinsic fluorescence, we can run label-free experiments. In MST, we have a tray that can hold up to 16 capillary tubes. In each tube, we put a constant amount of our fluorescent molecule, but we decrease the concentration of our other binding partner as we go on. This is called serial dilution. When we turn on our IR source, these molecules move away from the heated spot. The graph looks like this. All the molecules are equally dispersed initially and then we turn on the laser. After the laser is turned on, the fluorescence decreases as time goes on. IR source is turned off when the steady state is reached. Only one capillary tube is tested at a time. When the detector moves on to test the second capillary tube, we get another plot. We repeat this for 16 times. Don't you worry now, the instrument does not force you to run experiments 16 times each time. You can stop whenever you like. Ideally, you can end up with 16 curls or less. Highest concentration is at the top and lowest concentration is at the bottom because unbound molecules move faster and further away than the bound molecules. Then we take the fluorescence intensity from each curve at 20 seconds and plot another graph. We choose 20 seconds because this is when the sample usually reaches the steady state. The second plot looks like this. From this curve, we can determine the equilibrium dissociation constant. There's a long equation, but I did not write it here. On the other hand, the instrument calculates this value for you. The smaller the Kd value, the greater the binding affinity of the ligand for its target and vice versa. There are so many advantages of using this instrument. First, we do not have to use buffered salt solutions that can't provide accurate information about the binding behavior inside the human body. These experiments require only a few microliters. Yes, it is enough to run 16 experiments. These experiments are contact-free, so it avoids any room for contamination of the sample. MST allows measurement of interactions directly in solution without the need of immobilization to a surface. MST instrument is basically maintenance free. So yeah, you should try this.